Now we're going to look at meiosis and kind of go through the parts and the phases of meiosis. And there are some similarities, there are quite a few similarities to mitosis, but remember the point of mitosis was to make identical daughter cells as far as the number of chromosomes is concerned, whereas in meiosis we're trying to split the number of chromosomes. And that's going to happen in the first part of meiosis, which is called meiosis 1. That's where the number of chromosomes is halved, is split in half. And so meiosis 1 is kind of where it gets a little bit weird as far as what happens during the different phases. In meiosis 2, that is very similar to mitosis. So what happens to each of the chromosomes and the cell is going to be very similar to mitosis. So in meiosis 1, with the number of chromosomes being halved, the reason that occurs is because you've got these homologous chromosomes, these homologous pairs in the cell, and they're going to separate and kind of split in half. So the reason the number of chromosomes is halved in meiosis 1 is because during anaphase 1, when the chromosomes pull apart, it's the homologous pairs that separate. In meiosis 2, which is similar to mitosis, it's the sister chromatids that separate during anaphase 2. And we'll go into this in just a little bit more detail, but that's kind of the big picture thing you need to remember is that meiosis 1 is when the number of chromosomes is halved, and that's because the homologous pairs separate during anaphase 1. And then in meiosis 2, which is very similar to mitosis, the sister chromatids separate during anaphase 2. Okay, so let's kind of go into each of the details here. So these pictures show the series of meiosis 1. All right, so this is meiosis 1. And here is prophase 1. So this is, in this particular cell, there are 4 chromosomes. So the 2n number is 4. If this were a human cell, the 2n number is 46. There'd be 46 chromosomes, but that gets very complicated to show in a picture. So we're going to show a very simple organism that just has 4 chromosomes total. So 2 of these came from dad. The 2 blue ones came from dad. The 2 pink ones came from mom. And remember that each one you get from your dad and each one you get from your mom matches up, so you've got a homologous pair. So here's one homologous pair of chromosomes, the larger X's, and then the smaller X's would be a second homologous pair of chromosomes. Each of those chromosomes has a duplicated that occurs before prophase 1 ever happens, which is why it looks like an X. Each of these looks like an X. So there are 1, 2, 3, 4, 5, 6, 7, 8 sister chromatids total in this cell, in this prophase 1 cell. So you've got a total of 4 chromosomes in this cell, and that means that before they replicated, that would have been 4 chromatids, but after they replicated, there are 8. So 4 chromosomes, 2 that came from your dad, 2 that came from your mom, 4 total. So the 2n number is 4. After they've replicated, which happens before prophase 1 ever occurs, you've got a total of 8 sister chromatids. In prophase 1, the same thing's going to happen as in prophase of meiosis or mitosis, which is the nuclear envelope's going to break down so that these chromosomes can kind of float around the cell. Now, in metaphase 1, it is kind of interesting what occurs here because basically the homologous pairs line up on the metaphase plate. So they line up in the middle of the cell. So when we were looking at mitosis, let me scroll back down here to mitosis, notice that the chromosomes lined up kind of in a straight line, straight down the middle, okay, versus them lining up in pairs during metaphase 1. And these pairs that are lining up, these are homologous pairs. Remember, these are the larger X's, where the blue one came from your dad and the pink one came from your mom. And then the smaller X's, the pink one came from your mom and the blue one came from your dad. So they're lining up, the homologous pairs are lining up side by side on the metaphase plate. And this is going to be important because what it means is that the microtubules, the way the microtubules are attaching, when they start to pull these things apart, they're actually going to pull the homologous pairs apart during anaphase 1. So as these homologous pairs line up side by side on the metaphase plate, there's kind of this weird occurrence called crossing over that occurs. And what happens is, and you can kind of see this, because these pairs are so closely associated, they can actually exchange bits and pieces. So some of the genes that would have been from your mom, from the pink chromosome, could actually cross over to the blue chromosome and kind of mishmash the chromosomes. And so this is one way that genetic variation is introduced during meiosis, is through this process called crossing over. The other thing that is important to realize is that the way the homologous pairs line up is completely random. And so that term, which is random alignment, is another contributing factor to genetic variation. So in this particular example, the chromosome from dad, the blue one, is on the left-hand side, and the chromosome from mom, the pink one, is on the right-hand side. But that could be flip-flopped, like down here, where mom, the pink one's on the left, and the blue one from dad is on the right. And so this doesn't matter at all. The order of the chromosomes can be different every single time meiosis takes place. And so as these separate and go into the daughter cells, what that means is that each daughter cell is going to have a different combination of which chromosomes originally came from dad and which ones originally came from mom. And that allows us to have a lot of genetic variation in our offspring. In anaphase 1, the homologous pairs separate. And this is where the chromosome number is going to be halved. Because each of these pairs separates, what's going to happen is now one side of the cell is going to have basically the 1n number. So this is kind of where the 2n goes to 1n, is in anaphase 1 as these homologous pairs separate. In telophase 1, the new nuclear envelopes form around the new daughter nuclei. And notice that each of these daughter nuclei, where they separate in cytokinesis into two cells, only has 2 chromosomes. Okay, so it's got one big X and one little x, and then over here, one big X and one little x, whereas in the original cell, we had 2 big X's and 2 little x's. So this is why we're going from the 2n number, which was a total of 4 chromosomes, down to the 1n number with a total of 2 chromosomes in each cell. All right, so in meiosis 1, the 2 daughter cells that you end up with each have half the number of chromosomes of the parent cell. They've got 2 different chromosomes. But realize that these chromosomes are still made up of 2 sister chromatids that are paired together. So you've still got 4 chromatids in each of these cells, but only 2 different chromosomes. In meiosis 2, which is the next part, you're starting out with these daughter cells that have 2 chromosomes each. For this particular example, remember this was a human, this would be 23 chromosomes, not 2, but for this particular organism, the N number is 2 because the 2n number was 4. So the daughter cells have 2 chromosomes each. In metaphase 2, they're going to line up in the middle of the cell called the metaphase plate. Notice that it's a single layer again, kind of like what happens in mitosis, and that the centromeres, which are what join the sister chromatids together, the centromeres are attached to the microtubules, just like in metaphase of mitosis. And then in anaphase 2 of meiosis, these sister chromatids are pulled apart. And this is just like in mitosis. Sister chromatids are pulled apart. In meiosis 1, in anaphase 1, the homologous pairs have separated. In anaphase 2, the sister chromatids separate. And so when we get down to telophase 2, we have new nuclear envelopes forming around each of these daughter cells. And then after cytokinesis, you end up with a total of 4 daughter cells. And each of these daughter cells has 2 chromosomes. And the chromosomes they have are all different. So each of the daughter cells is different from each other and from the parent cell. You got a blue chromosome that originally came from dad and a pink chromosome that originally came from mom in each of these cells. But notice here, because of crossing over, the bits and pieces kind of were exchanged. So all of these kind of have a different genetic makeup from the parent cell, which is why you can look very similar to your parents or quite different from your parents based off of how these genes kind of recombine. The last thing I want to go over real quick is the cell cycle. Remember that mitosis or meiosis is only a very small part of the cell cycle itself. And so in the eukaryotic cell cycle, there are a series of phases. In the G1 phase, which is also called the gap1 phase, this is where organelles are going to duplicate and the cell is going to grow in preparation for mitosis. There are some cells that don't divide, or at least divide very infrequently. Examples of that would be your brain cells or your heart cells, where if there's damage, it's very difficult for them to heal. So G1 is organelles duplicate and the cell grows. In the S phase, that's the synthesis phase, DNA is copied. And so it's replicated, getting ready for mitosis. And in the G2 phase, that's another growth phase. There is some protein synthesis to get ready for mitosis and just some more cell growth. And then finally, in the M phase, in mitosis, that's where the cell actually duplicates itself. All right, so when we're talking about DNA replication, DNA copying, a couple things you need to keep in mind. It's termed semi-conservative because when you have this original strand of DNA, it's going to be split in half, and a new strand, which here is black, a new strand is going to be built around the old parent strand. And it uses those base pairing rules that we talked about before, where A binds with T and C binds with G. It uses those base pairing rules to make the new strand of DNA, but it's termed semi-conservative because when you're done, you've got one old parental strand and one new strand, making up each daughter piece of DNA. The enzyme DNA polymerase is the enzyme that actually does all of this. So DNA polymerase splits the DNA molecule in half and helps join the new nucleotides to that. So DNA polymerase is an important enzyme to remember because that's involved in this DNA replication. As far as the cell cycle goes, there are some checkpoints, and those checkpoints help prevent cancer. And they occur at different points throughout the cell cycle, and your textbook kind of goes over that. Make sure you understand, for the purposes of the exam, proto-oncogenes, oncogenes, tumor suppressor genes, and specifically, as far as tumor suppressor genes go, the P53 gene. All of those things will be on the exam, so make sure you understand what those terms mean and how they relate to cancer. The other thing you should know for the purposes of the exam is how to diagnose cancer. And we talked about fine needle aspirates as well as surgical biopsies. And then radiographs or x-rays can also help you diagnose cancer. As far as treatment of cancer, there is chemotherapy, which involves using chemicals to kill off the cancer cells in your body. There's radiation therapy. And then there's also surgical therapy. So surgically removing the tumor. If you have any questions about this, make sure you email me about that. Also, make sure you understand the table that's on page 115 and 116 in your textbook. That's really important as well. There will be several questions from that table involving types of cancer, common ones found in young men, the most likely way you're going to die of cancer, that sort of thing. So make sure you understand and memorize what's in that table.